My research is about metabolites in the ecosystem. Metabolites are a group of small molecules you can find in nearly all living organisms. It can not only indicate us how, if a planet is under stress, if an ecosystem is under change, it can also inspire us to making fertilizers, biostimulators, biopesticides as a nature-based solution. I did most of my work in American Southwest drylands, where I looked at two different grasses, Arizona corntop and lemon grass, and tracked the metabolite they produce to the soil and how these metabolites have an effect on the microbes associated with them. And a small subset, but an important part of that is iron, which is kind of like the energy drink for microbes. However, it is not very bioavailable. And one of my colleagues, her work is focusing on how microbes are producing a specific type of metabolite called siderophore, which can increase the bioavailability of iron from the soil.
My work in the Twiley lab is looking at how these desert organisms have adapted to survive in these really harsh conditions in the Sonoran Desert. One way that they do that is by producing siderophores, which are metabolites that can very tightly bind iron that is usually not bioavailable in the soil, and then bring it back into themselves. So to investigate these siderophores and these siderophore-producing organisms, what we typically do is we start by growing them on agar plates. And once they've grown, we do colorimetric assays called an OCAS assay. So what this is, it is a dye that is typically blue when it has iron bound to the dye. And if the dye sees a siderophore, it will give up the iron that's bound to it to the siderophore with the higher preference for that iron. This causes a color shift in the dye from blue to yellow, and we can use that to confirm that we actually have siderophore activity being done by these bacteria. Or this can have potential agricultural applications, as far as siderophores have been shown to help promote plant growth and compete against other bacteria and pathogens that are typically not very helpful for the organisms surrounding it. Medically, we can also use these to potentially bind excess iron in the blood and remove it from the body.
I'm trying to capture all those very tiny and small signals, simply like microbes responding to the environment, to inform us about how microbes doing in the soil. So provide us some tiny thing we can try to manipulate and we can utilizing microbes for our own purposes.